How does DNA lead to traits? DNA is translated into traits, or something is called proteins. Actually, traits are made of proteins. How does the DNA lead to traits? We have genes in our DNAs, and how these genes are translated or turned into traits. The central dogma is a theory of molecular biology states that information flows in one direction, from DNA to RNA to protein. And protein here means traits. DNA to RNA to protein. Not DNA to traits directly, but DNA to RNA to protein. As we know, DNA carries our genetic materials, and our genetic materials converted into traits or proteins. Why DNA is converted first to RNA and then protein? Because to convert the DNA into protein, you have to get the DNA out from nucleus. But the DNA is so large, very large molecule, and can't pass outside. That's why we have to transcribe or make another copy in a single strand is called RNA from a gene that is required to be translated, and take this strand from the nucleus to outside to be translated. To convert our genes that are found in our DNAs to traits or to proteins, DNA must be converted into RNA and then to protein. And protein means traits. This process is called protein synthesis. Protein synthesis. Protein synthesis is a process that uses DNA to create RNA that is called ribonucleic acid that is used in protein production. Protein synthesis has two steps. Number one, transcription. Transcription. And it occurs in the nucleus to convert a gene into RNA. To convert a gene into RNA. Number two, translation. Translation. And it occurs outside of the nucleus. It occurs in the cytoplasm or ribosomes. And translation to convert RNA into protein, or to translate RNA into protein. And protein means here traits, our traits. What's the difference between RNA and DNA? RNA and DNA. At the beginning, DNA and RNA, they are both nucleic acids. And they are polymers. They have monomers or subunits are called nucleotides. DNA has a name, deoxyribonucleic acid. Deoxyribonucleic acid. While RNA is ribonucleic acid. As we know, DNA is named after its sugar. DNA has deoxyribose sugar. RNA named after its sugar. Guess what is the sugar type of RNA? RNA is called ribonucleic acid. What is the type of sugar of RNA? Deoxyribose or ribose? Yes, ribose sugar. RNA has ribose sugar, not deoxyribose. That's why RNA is called ribonucleic acid. Another difference here, DNA has two strands. While RNA has only one strand. One strand. Another difference, DNA has 4 nucleotides. And RNA has as well 4 nucleotides. But DNA has A, T, C, G, or adenine, thymine, cytosine, guanine. And they have same phosphate sugar, but they differ in nitrogen base. In RNA, it has 4 nucleotides: adenine, cytosine, guanine, and uracil. Where is the T? Where is the thymine? No thymine in RNA. No thymine in RNA. It has another nucleotide, is called uracil. Uracil is in a state of thymine. So, RNA has 4 nucleotides: A, U, C, G. A, U, C, G. Adenine, uracil, cytosine, guanine. While DNA has A, T, C, G. Adenine, thymine, cytosine, guanine. Okay, here is a chart of characteristics found in either DNA or RNA or both. Use the check marks to indicate which are found in DNA and which are found in RNA. Number 1, ribose sugar, found in RNA. Number 2, deoxyribose sugar, found in, of course, in DNA. Oh, phosphate. We know DNA and RNA are nucleic acid, and they have nucleotides. And all nucleotides have the same phosphate group and sugar, and they differ in nitrogen bases. DNA and RNA have phosphate group. So, phosphate present in both DNA and RNA. Adenine present in both. Thymine, which indicates T, found in DNA, not found in RNA. Uracil, U, present where? In RNA, not found in DNA, only found in RNA. Uracil found in RNA. Guanine present in both cytosine present in both double-stranded. Which one is double-stranded? DNA, because it has 2 strands. Single-stranded. Which one is single-stranded? Yes, RNA, because it has only 1 strand. Which one remains in the nucleus? Yes, DNA, because DNA is so large, very, very big molecule, can't pass outside. DNA in the nucleus. Which one moves out of the nucleus? Of course, RNA. RNA can move outside, while DNA can't move outside. Transcription. Transcription is the first process of protein synthesis. It happens inside the nucleus to make RNA from the DNA. To convert only one gene into mRNA. Convert a gene or transcribe or copy a gene into mRNA. mRNA here means messenger RNA. Messenger RNA because it carries the message from the DNA. It carries the message from the DNA. In transcription, RNA polymerase enzyme is used. RNA polymerase enzyme is used to make RNA. In picture here, you can see 2 strands of DNA and transcription unit. Transcription unit refers to the gene that will be transcribed into mRNA. Before transcription unit, you can see in the DNA itself, start site, a start site. It is called promoter. Promoter. A promoter is a start site of a gene. What is the function of that promoter or that start point or that start site? Promoter here to tell the RNA polymerase to come and start from this point. When the RNA polymerase binds to the promoter, the DNA strand begins to unwind. We need here only 1 strand from the DNA, not the 2 strands. Only 1 strand of the DNA is going to be transcribed into messenger RNA. The second step of transcription, after unwinding the DNA strand, you can see here in the picture, DNA strands are blue and the RNA that is formed is red. After the RNA polymerase binds to the promoter, it's going to move through the strand to add new nucleotides that are complementary to one side of the DNA molecule. Are complementary to one side of the DNA molecule. What does it mean? In the picture here, from the right side, you can see from the right side, A in DNA takes U. Why? Why it didn't take T? Because we are forming RNA. We are forming RNA, not DNA strand. RNA strand has no thymine. It has uracil instead of thymine. Nucleotide A takes U or pairs with U. The second nucleotide A pairs with U. Third nucleotide G pairs with C. A pairs with U. Then G pairs with C. C pairs with G. T pairs with A. A pairs with T. No, pairs with U because we are forming RNA. And so on. RNA is formed by using RNA polymerase enzyme that added nucleotides or RNA nucleotides that are complementary to the one side of DNA molecule. Questions. For example, DNA sequence A, T, T, C, G, T, A. The complementary mRNA, what is the complementary mRNA here? To form mRNA from this DNA sequence, what we are going to write? Okay, A pairs with U because we are going to form mRNA, not DNA. A pairs with U. T pairs with A. No problem here. T pairs with A. C pairs with G. G pairs with C as well. T pairs with A. A pairs with A. A pair with U. So, the answer, U, A, G, C, A, U. The second question, here, the given mRNA is UAUCGAU. Can you make DNA from this mRNA? We are going to make DNA. Be careful. We are going to make DNA. You will take A. You will take A. And A takes U? No, it is a DNA. A takes T, thymine, because the DNA has no uracil. It has thymine, T, instead of uracil. A takes T, U, A. C, G, C as well. A, T, U, A. So, the complementary DNA will be A, T, A, G, C, T, A. There are three main types of RNA. Transfer RNA or tRNA, transfer RNA. Second type, messenger RNA or mRNA. Third type ribosomal RNA or R small RNA. Transfer RNA, it carries amino acid, specific amino acid, to the ribosome to make protein. It carries amino acid. Messenger RNA, it carries the message from the DNA. It carries the message from the DNA. It carries a message from the nucleus to the ribosome. Messenger RNA is formed inside the nucleus and it can leave the nucleus to the ribosome outside in the cytoplasm. Ribosomal RNA, it makes up ribosome. Ribosome is made up of ribosomal RNA and protein. Ribosomal RNA is a component of the ribosome. Ribosome has two components, ribosomal RNA and protein. Messenger RNA. Messenger RNA, it carries instructions or the message from the DNA. The DNA is located inside the nucleus. Messenger RNA takes the message from DNA and it gets out of the nucleus and it goes into cytoplasm or ribosome. Messenger RNA carries a message as specific nucleotides or sequence of nucleotides. Here in the picture, it shows examples of sequence of RNA nucleotides that indicate the message of DNA. A, U, G, A, C. How can we read messenger RNA? It is a message. How can we read it? Here we have nucleotides. H3 nucleotides, for example, A, U, G, they form codon. A codon. A codon has 3 nucleotides. H3 nucleotide form a codon that gives 1 amino acid. What's the meaning of amino acid? Amino acid is a component or the monomer of protein. Is the monomer of protein. To form a protein, we need amino acids. That's why nucleotides here give amino acids. But not only one nucleotide gives 1 amino acid, no. H3 nucleotide. H3 nucleotide gives 1 amino acid. 3 nucleotides make a codon which gives 1 amino acid. Here in this picture, can you guess how many amino acids produced from this strand? How many amino acids? Yes, 2. Because H3 is going to H3 nucleotide going to make 1 amino acid. 1 amino acid. Transfer RNA. Transfer RNA or tRNA. Transfer RNA, it carries amino acids. It carries amino acids to the ribosome. You can see here the amino acid is colored with green. tRNA carries amino acid and it has sequence of nucleotides. It is called anticodon. Anticodon, which is complementary to a specific codon of mRNA or messenger RNA. Anticodon, complementary to a specific codon of mRNA. In the previous slide, we had strand of mRNA, A, U, G, A, C, and a codon had 3 nucleotides. The first codon was AUG. What is the anticodon of this codon? What is anticodon of tRNA to AUG of mRNA? AUG, A pairs with U, U pairs with A, and G pairs with C. Ribosomal RNA. Ribosomal RNA, it makes up ribosome. It is a component of ribosome. Ribosome has 2 components, ribosomal RNA and protein. The ribosome itself is a site of translation. Is the site of translation that will be explained in the next PowerPoint. Ribosome is made up of RNA and protein. This picture shows ribosome. Ribosome. This picture shows a quick review of the second step of protein synthesis, that is translation. In translation, we use the 3 components of RNA. We use mRNA, the strand, and tRNA, and ribosome. As you can see here, ribosome has 2 subunits, blue and brown. One large subunit blue and small subunit brown. It attaches to the strand of mRNA. And tRNA, which carries amino acids and has anticodon that complementary to codons of mRNA, they are landed in ribosome. tRNA carries amino acids and has anticodon that complementary to a specific codon of mRNA. From the right side, we have tRNA carries red amino acid and has anticodon U, which is complementary to codon A. The second tRNA, which carries amino acid with blue color, has anticodon AG that is complementary to UC. The meaning of UC here is a codon in mRNA. And so on. tRNA bring amino acid to form protein.